Now I'm going to introduce Michael. Okay. Okay, Michael Platt is director of the Duke Institute of Brain Sciences and director of the Center of Cognitive Neuroscience, professor of neurobiology, evolutionary anthropology, psychology, and neuroscience. Boy, that's a lot of names. So I became familiar with Michael's work when I was a postdoc, and immediately the work impressed me. It was one of the first studies I became aware of where you could measure social valuation using drops of juice and eye movements. I mean, that was really cool. It was a new idea to me, and I was very impressed with the work. I thought it was creative, and I recommend his papers to my students to read. So a very influential person in neuroscience and neuroeconomics. So just to finish, Michael has an illustrious career, many prizes, and in particular, he's well known in the general media and has, has his work featured in New York Times, Washington Post, so I think maybe his name is well known among the general public. Would you say that? No. Okay, without further ado, Michael Platt.
Thanks, Anna. So, no, I don't think I'm particularly famous or anything like that. So, but it is a pleasure to be here, and I want to thank the Kavli Foundation and our partner institutions for helping put this on, and in particular, Mu Ming for hosting us here. It's been really fun visit, and it's a wonderful venue. So, congratulations on the building and everything you're building here. And you commented on the title for this symposium in general, which you found to be interesting. It's actually what our provost said about the title. He said, that's a really interesting title, Building Interest and Capacity. And I think it's actually really appropriate because it seems already we've built interest. I mean, look at all the people here. And from all my discussions over today and the past couple of days, it's quite clear that capacity is well on its way. And so great things are going to happen here. So let's get right into it. We're going to talk about social behavior. So the question I want to put to all of you is, why are you here? Why are we all here? I don't mean that in a religious or cosmic sense, but what are we doing here? And the reason we're here is because we come together to learn from each other. We come to make friends, to make alliances, to form partnerships, and when we do so, we can often achieve greater good, greater ends than we might achieve on our own. This is something that's really special about the human species. On the other hand, a lot of the reasons that we do this are motivated by competitive reasons. So not many people here live in North Carolina, but these are the two coaches, Mike Krzyzewski and, now I'm blanking, Roy Williams. I don't know why I just totally blanked because I hate the guy, but even though he only lives about 5 miles from me. So these are the two coaches of the men's basketball teams for Duke and the University of North Carolina, separated by 9 miles, and this is one of the most intense rivalries in sports. And so we are just, as a species, not only do we cooperate, but we're incredibly competitive. And these are two tensions that kind of rule our lives and I think rule our brains. And over the last 10 or 15 years or so, and I think a lot of this work has been done under the auspices of neuroeconomics, we've learned a lot about the twin forces, the mechanisms that mediate these processes of sort of cooperation, caring about others, prosocial behavior on the one hand, and on competitive behavior on the other. And most of these studies, most of what we've learned has been gleaned from imaging, fMRI studies done in people, right? Now, that's fantastic. It can reveal a lot about the brain areas that might contribute to a particular process. But as we all know, the temporal and spatial resolution at the moment are not quite precise enough to give us very good mechanistic insight into the local computations by individual neurons, the mechanisms by which these systems might actually play out. And so what we do is we turn to animal models in which we can make the kinds of much more direct, precise, invasive measurements that allow us to understand how neurons in the brain contribute to these various behaviors. And in addition, we can also manipulate these systems quite directly. So we can turn neurons on and off. We can modulate the neurochemical milieu and in that way see how the system might function. In particular, we, in my lab, focus on rhesus macaques as a very good model for the decisions that we make, and in particular the kinds of decisions that we make in social contexts. So these critters are really ideal for that in that regard. They are both very competitive, so they are strongly oriented toward achieving status and acquiring resources, but at the same time, they, like us, form strong social bonds with each other. They make friends, they make alliances. They seem to assess the value of other individuals for making a friend as someone who can help them in the future, and they also learn from each other. In addition, we know that the brains of these animals are organized in very much the same way as our own, and that a lot of the underlying biology, the genes, the proteins, et cetera, that go into building these brains are shared with our own. In addition to that, I just want to point out the fact that one of the great reasons, in addition to being able to measure, manipulate, et cetera, the brains of these animals, is that by understanding how they work in their own context, they give us insight into the toolkit of computational motifs and neural circuit mechanisms that evolution can apply to our own brains, that we carry along with us in our own heads. So I think a lot of what we take to be kind of biases, irrational decisions that we make in a context like a great city like Shanghai probably arise from the fact that we're applying what actually evolved in a critter like a rhesus macaque some many tens of millions of years ago. Okay, so how do we actually do this in the lab? What we do is we try to come to some understanding of a particular behavioral problem, like how do monkeys make friends? We try to turn that into a problem that can be applied in the laboratory in a way that preserves some of the natural, the aspects of the natural behavior, but is also rigorous and controlled and repeatable and suitable for neurobiological study. And then we do things like make extracellular measurements of the spiking of neurons. We often collaborate with other folks who are doing brain imaging studies in humans to see what kind of inferences we can draw between what we observe at the cellular level in monkeys and what we can see at the level of hemodynamic activation in humans. We also have begun to use pharmacological studies to try to tweak the system. And then finally, most recently, we've begun to go back into much more natural decisions. And in this case, this is the pattern of relationships, affiliative or positive social relationships between individuals in a group of monkeys on an island who are freely moving, living naturally. And we are studying these animals, how they make decisions in the wild, and how their underlying genetic and other aspects of their biology shape those behaviors. And the idea is to kind of take this full circle, in the end, perhaps selecting animals from this kind of population based on their genotype and their phenotype and beginning to understand, but to use our neurobiological tools in the laboratory to unpack that system. Okay, so let's turn to positive social behaviors. What are the neural mechanisms that actually motivate us to attend to other individuals, to learn about them, and in particular, to take into account their experiences to motivate our own behavior? This is something we've been interested in for a long time, and we studied it for probably about almost a decade in terms of very simple decisions about whether or not to look at another individual to get information about them. But now what we've done is we've begun to look at this in an actual social interaction. So this is a variant on a very simple dictator economic game where we basically have two monkeys now who are sitting in the experimental room. Each monkey sitting in a primate chair. They're more or less catty-cornered to each other. Each monkey has a little computer screen in front of him. Each monkey has a juice tube in his mouth. We will typically be recording from or otherwise manipulating neuronal activity in one of these monkeys. I'll refer to him as either the actor or the donor. And then in this case, there's another monkey who's basically a passive recipient. Okay? And in this first study, what we did was we taught these monkeys that there were different sets of cues, shapes, colored shapes on the screen that led to different kinds of outcomes. So there might be one set of cues that would, after they were displayed, would result in a squirt of juice going to the actor monkey. Okay, that's not very unusual. Another set of cues would, after a delay, result in a squirt of juice going to the recipient monkey. Another set of cues would result in juice going to both monkeys. That's hooray, we all got rewards. And then finally, another set of cues that would result in nobody getting a reward. Okay? And this is just sort of a sequence of how those trials play out. Now, once they've learned the meaning of these different sets of cues, we then give the actor monkey choices between cues that are matched for the payoff to himself. And I think the most interesting one for our purposes is a choice between a cue that had predicted a reward to the other monkey, or a reward to nobody. So, yes?
You have to know that the other
Yeah, well, what monkeys know and don't know about their worlds and the experience of others is, I think, a really interesting question. I'll show you in a minute that their behavior betrays that they distinguish between those outcomes. And we also monitor the eye movements of these monkeys, and we see that that monkey, the actor monkey, watches the other monkey drink the juice whenever he does get a squirt of juice. So from that, we infer that he understands something about it. So this is a situation in which the actor monkey had never received a squirt of juice in association with either of those cues over the many thousands of trials that he had experienced. Some people might even argue that our actor monkeys would refuse to make a choice, refuse to play the game under those conditions. If the monkey, however, expresses a preference for one or the other of those two cues, that suggests that he's actually incorporating some information about the experience of the other animal, the social reward, if you will, into his own experience that drives his behavior. The other set of choice that we give monkeys that's of interest is a choice between a reward to self and a reward to both of us. In that case, both of those sets of cues has always resulted in a reward to the actor monkey, so you might expect him to be indifferent. He's just always getting a reward. If he shows a preference for one or the other of those outcomes, that also shows us that the social context of that reward is having an impact on his internal processing of the value of that reward. Now, what I'm plotting here is the result of a number of different sessions in which we tested two actor monkeys against a number of different recipient monkeys. This is the context that, for me, is the most exciting. It's a choice between reward for the other guy, reward for nobody. This is the other context, which is a reward for me, a reward for both of us. And this is an index of what we call the vicarious reward. So bars that are going up are more prosocial choices. Bars that are going down are more selfish or antisocial choices. And in this context, we can see that our actor monkeys tend to be very prosocial. This is a logarithmic scale. So they're favoring about 3 to 1. Reward the other monkey versus reward nobody. Already a very interesting outcome in this task. They're not indifferent. Now, you might worry that, in fact, what's going on here is they just like the sight of juice coming out of a tube. These are monkeys, of course, who are on controlled access to fluids. And so it's important to show that in the absence of a social partner here, we don't see that kind of preference. And in fact, here, there's this tiny little red bar here. And what that refers to as our non-social control, in which we remove the other monkey, remove the recipient, and replace him with a bottle that collects the juice. And we can see that into that condition, our actor monkeys are indifferent. So this preference that we're seeing here expressed is something that's fundamentally social in nature. It depends on the other monkey. Now, we see this weird, you might consider, if you're an economist, preference reversal, in the sense that now when it's the choice between my own reward or both our rewards, our monkeys are predominantly selfish. So they really seem to not like the experience of drinking juice while another monkey is drinking juice at the same time. I think that some of us can identify with that feeling, that sometimes a reward is not as special if you see somebody else having it at the same time. I think for our monkeys, I mean, our intuition is that they feel that this is a competitive context, that they might be drinking from the same puddle, if you will. That's a complete speculation. One of the ways we're trying to actually test that idea is by giving monkeys now choices between quantized pellets of juice that they can actually see what each monkey is getting and know that the other monkey is not getting more than him. The important point here, though, to take home is that there are fundamentally socially influenced preferences in this task. Now, I can go one step further and show you that the relationship between the two monkeys has an impact on the value of these outcomes. So if we compare monkeys who are, based on their familiarity, monkeys who are more familiar with each other, the actors tend to be more giving. Okay, it's more rewarding to give reward to the other monkey than if they're less familiar. And it's a little bit less competitive. And also the sort of dominance relationship between those two monkeys also has an impact on the social reward value. So monkeys, actor monkeys who are dominant to the recipient tend to be a little bit more prosocial and a little bit less competitive than the other way around. I mean, I think there are a variety of explanations that we might entertain for that behavior. So how does this actually play out in the brain? I think that for a set of behaviors like this, it would be very interesting to trace out the transformations that occur from kind of the initial processing, I'm sorry, I'm losing this, processing of information about who is out there in the immediate social environment and what are they doing and what are they experiencing, which occurs in the temporal cortex. And the eventual translation of that information into, thank you, information about, all right, information about the value of those options, and then ultimately translation into motor commands that express the behavior. So we could really start anywhere in this process. We elected to go kind of right into the meat of things in the frontal cortex. This is a coronal section through half of the monkey brain. And in an initial study, we studied 3 different areas, which you've heard about today. The orbital frontal cortex, and both the dorsal anterior cingulate cortex and the anterior cingulate gyrus. These are all areas that have been implicated in various aspects of decision-making, learning, and reward processing. And the anterior cingulate gyrus is of particular interest. It's often referred to as the medial prefrontal cortex in humans. It's an area that's been associated with empathy, theory of mind, and some information processing about the experiences of other individuals. Now, what we found in these areas was pretty striking. These are the responses of individual neurons from the OFC, the dorsal anterior cingulate, and the anterior cingulate gyrus, but they basically look like the population of neurons that we recorded from, and the color codes here referred to the outcomes that we're segregating neuronal activity by. And the main point here is that the orbital frontal cortex neurons show a very strong, sustained response anytime the actor monkey chose to give himself a reward. So anytime he got his own personal, direct, egocentric squirt of juice, these neurons were strongly activated. Okay? If he did not get his own juice reward, we saw that that activity was basically shut down. By contrast, neurons in the dorsal anterior cingulate cortex showed the opposite pattern of response. Almost no activity if the monkey got his own reward. Strong activity anytime he decided to forego a reward, anytime he did not get a reward. So we tend to think of this kind of response here as a counterfactual or fictive signal about what could have been had the monkey acted differently. What kinds of rewards are there out there in the environment that I did not receive? And we had shown in a previous study in a non-social decision-making task that neurons in the dorsal anterior cingulate cortex convey information about counterfactual rewards that the monkey can use to learn and update his behavior on subsequent decisions. Now, the neurons in the anterior cingulate gyrus turned out to be the most interesting, and there are 2 flavors of neurons there. The most predominant are neurons that basically only distinguish between nobody getting a reward and somebody getting a reward. So these neurons respond the same way whether the actor monkey gets a reward himself or gives a reward to another monkey. You can think of these neurons, if you will, as mirror neurons for reward. So they encode the experience of another monkey in direct terms of the experience of the donor monkey himself. There's another flavor of neuron that we recorded from this area as well. These neurons were selective for the experience of another individual. They did not respond either for self-reward or for nobody getting reward. So these are very interesting because they seem like the building blocks for incorporating the experiences of another individual into one's own model of the world and using that information to drive behavior. Now, I'd like to be able to show you that we have functional evidence demonstrating that these neurons actually underlie the behavior of the monkeys in this task. We haven't completed that experiment yet, but we've done an experiment that's closely related, and this is an experiment in which observer monkeys are watching a demonstrator monkey taste different kinds of foods that he's never seen before. So we basically cook up little gelatin pearls that have arbitrary relationships between their color and their flavor. So they can be sweet or bitter, and they can be any color. And the monkeys basically learn very rapidly, either from their own experience sampling the foods, which is good and which is bad, and they also learn equally well from watching another monkey taste the foods. So see the other monkey pick up a piece of food and spit it out and throw it away, they won't select that piece of food. If they see the other monkey pick one up and eat it and consume it, then they start licking their lips and they want to eat it on the next trial. So what we've done here is use mucimol injections, GABA agonists, to inactivate neurons either in the anterior cingulate gyrus or in the insula for control. And we've looked at the ability of the monkeys to learn from their own direct experience of tasting the foods versus learning from watching another monkey. And what we do is we reverse the contingencies between the color and the flavor that it's paired with right here at this boundary. And what we see here is that our monkeys, when we inactivate the anterior cingulate gyrus, they have no problem making this switch based on their own experience of the flavors, but they can't learn from watching the other monkey. It's as if they cannot incorporate the experience of the other animal into their own model of the world, their own model of the association between color and flavor. Inactivating the insula had the exact opposite outcome. So we saw no impairment in social learning, no impairment of learning from watching another individual, but we see an impairment in learning from direct experience, which is not unexpected given the role of the insula in representing gustatory information, in particular bad tastes. So this suggests that when monkeys and presumably humans are making decisions in a social context, that there's a suite of brain areas, a network of brain areas in the frontal cortex that becomes activated in very specific ways, and that there's one particular area that seems to be very specialized for incorporating and representing information about the experiences of other individuals, an empathy-like signal, if you will, into the monkey's model environment, and this helps to motivate his behavior in subsequent trials. Okay, I'm going to turn to the other question now, which is one of competition. Of course, competition is really interesting because it promotes strategic, dynamic, unpredictable, innovative behavior. We saw that from a couple of the talks today, which talked about matching pennies and the odds-evens game when monkeys can become very unpredictable. Those studies, as you heard, were not done in social contexts, and so we wanted to see what kinds of behavior and what kinds of, which neural circuits might be involved in actually mediating strategic, social, competitive behavior. And what we did was to adapt a very simple version of a penalty kick game for play between pairs of monkeys. So it's basically the same kind of setup that I described to you before. Two monkeys seated right next to each other. Each of the monkeys is looking at a computer screen, but in this case they're going to be using joysticks to play a game against each other. And so one monkey is going to be assuming the role of the penalty kicker, and the other monkey is going to be the goalie. If the penalty kicker can get the ball past the goalie, he gets a squirt of juice. If the goalie can block the ball, he gets a squirt of juice. And we titrate the size of the goalie so that the payoffs of the two monkeys are roughly equal. And so here's a video showing a basic outline here. So here's the goalie, and here's the kicker. And you can see, so the goalie blocked the ball that time, he got a squirt of juice. And you can see that they can adopt many different paths here. They have many degrees of freedom. So he won that one. See, this is great. It's a great talk to have at 4 p.m. because we can just watch videos. That's my favorite one. So if I, what's that? Yeah, it's the head fake. So if I didn't tell you these were monkeys, you might have thought they were my kids playing against each other. My kids actually play much more sophisticated video games, but Paul and I would have played games like this, I think, back in our day. So one of the really interesting things is that this is 1 day's worth of one monkey playing against a recording of a monkey from the day before. And when they're playing against a recording of a monkey, you can think of that recorded monkey as exhibiting the same kinds of temporal dynamics as a live monkey, but he's not reacting to the kicker. And you can see that, in fact, our kickers pick up on that very rapidly, and they just play a very stereotyped kick. Okay. By contrast, if we have our monkeys playing against a live goalie, we see that they seem to take a straight path until the very end, and then they execute a series of circuitous movements, which we call feints or fakes. Okay. And so these are two very statistically different patterns of behavior that we see. Our monkeys become very unpredictable, and we demonstrate that with a mutual information statistic and a variety of other measures. When they play against a live goalie, they're very unpredictable. Now, is this a socially driven behavior, or are they just kind of reacting to lights on the screen? What's that? This is like you're like a planted questioner right here. So here's the gaze points of our kicker monkey during one of these sessions. Here's the location of the monitor, and we can see that the monkey starts out looking a lot at where the ball starts and tracks the ball and the goalie, but he also spends a lot of time looking at his opponent's face and a lot of time looking at the other monkey's joystick. So it looks like he's trying to pick up on any cue possible to predict the other monkey's behavior. So that's one thing that tells me that there's something interesting going on there. Now, we're doing a lot of work now trying to figure out exactly what are the visual and other kinds of cues that our monkeys use to identify that they're playing against a live monkey. And I can talk about that later. But I did want to show you, I got very interested in this question about what are these monkeys really thinking about? Is this really a social problem? How are they approaching this? And so what I did one day, so that's me playing against a monkey. Now, it's my first day playing. He hadn't played in a few months. Sorry, I'm trying to skip this ahead. And he was basically doing a pretty good job against me. It turns out that I won, it was more like 55-45. Sorry. I didn't get any rewards, though. But I want to show you something really interesting. So let's see if we can watch the next couple of trials. He's got kind of a go-to move, that one. That's not it. I think I'm at the wrong. So that's really, that was an amazing moment for me because, I mean, I already felt that I was playing some sort of interesting, almost human, intelligent agent. But in that instance, I picked up on one of his go-to moves and I blocked him. And he got really angry. So if anybody who has experience with monkeys, this monkey anger. But what's interesting is he didn't get angry at the computer monitor. He didn't get angry. He didn't bite the juice tube. Instead, he turned and he threatened me as if I were the source of his misfortune. And so I think that's really, now that's completely anecdotal. I mean,
Does this make you submissive? Are you dominant or submissive?
Well, I'm always dominant. Anyway, I mean, it's just, so we're trying to figure out ways to use this kind of observation to get inside the head of this monkey and what is he really thinking and experiencing. It's an interesting proposition. But in terms of what they're actually doing, I think the really interesting question here is whether these kickers and the goalies are just reacting to each other. Is this just a rule-based play? Or does the kicker, for example, have an intention in mind? Does he actually have a goal from the very beginning of the trial, and then his behavior is, in a sense, misleading the other monkey by broadcasting inaccurate information about where he's going? And in fact, I'll show you two pieces of evidence that the latter part is the case. So the first is that the kickers look at their goal before they start. So the little red dot is the monkey's eye position, and the big yellow dots are where he's looking at the beginning of the trial. And you'll see that the ball almost always ends up where the monkey had looked at the beginning of the trial. He's doing a little look ahead that betrays his goal. And in fact, if our goalie monkeys could pick up on this, they could more effectively block the kicker. These two monkeys are seated kind of catty-corner to each other, so I think it's impossible for them to pick up on this. We now have some studies where we're beginning to look at monkeys who are actually face-on, and they might be able to pick up on some of these cues. This is just data showing that the correlation between eye position at the beginning of the trial and where the ball ends up, and you can see that there's a peak here for one single session. Across all our sessions, where the monkey looks at the beginning of the trial strongly predicts the end point of the ball. Okay, the other piece of evidence I'm going to show you actually comes from neurophysiological recordings. These are both done to kind of in a reverse inference way to help us intuit what's going on in the monkey's head, but also to provide some insight into the neural circuitry that mediates these kinds of unpredictable behavior and planning behavior. So if you look at the neuroimaging literature, there are a couple of areas that always pop up when people are kind of planning to mislead others, and those include the dorsolateral prefrontal cortex and dorsomedial prefrontal cortex, which are involved in a whole suite of different behaviors. But of course, the DLPFC plays a crucial role in planning ahead. And so, of course, we have put electrodes into the homologs of these areas in monkeys, and what we found is very interesting. So when monkeys are playing against a live opponent, we can see neurons like this one that become active in the very beginning of the trial when the monkey is going to make an upward faking movement. And it comes well in advance of him actually executing that movement, which is aligned here. They're not active for downward feints, straight movements, etc. We have control conditions in which we ask the monkeys to basically replicate the same trajectories of movement. So this is a control for just movement planning. And under these conditions, we don't see any strong neuronal activation. And in fact, if we look at the activity of neurons in the DLPFC, we can increase our ability to predict the monkey's behavior. He becomes much more predictable to us from the beginning of the trial. The dorsomedial prefrontal cortex neurons, on the other hand, don't have strong activity early in the trial. Their activity begins much later in the trial. It still seems to be selective for playing against a live opponent, but it seems to be provide much more information about the last movement the monkey will use to achieve his goal. So these two areas seem to carry very different sorts of information. And in a study in which to complete this study, we used mucimol injections again to inactivate either the dorsolateral prefrontal cortex or the dorsomedial prefrontal cortex, and we compared that with saline injections. And what we found is that the dorsolateral prefrontal cortex is critical for our kickers to execute these kind of unpredictable movements. So when we inject mucimol here in gray, sorry, I lost the scale thing here, the legend, our monkeys become much more predictable and the kickers lose the game more often if we inactivate the DLPFC, but we don't find that when we inactivate the dorsomedial prefrontal cortex. So again, what we find here is different. These aren't areas that we would consider to be specifically social in nature, but they seem to be critical for driving complex social interactions. So I'm going to now turn to a different set of questions in the last few minutes, which have been motivating a lot of our behavior, and this came up actually a couple of times today when we were learning about kind of stickiness for behavioral tendencies. So what is it that actually drives individual differences in social behavior? Some individuals are very gregarious and social. Other people are kind of much less social. This is well appreciated by comparing these two guys here. I hope they're familiar to everybody in this room. They're similar in a whole variety of ways. They're both computer nerds. They're both fantastically rich. But they differ in at least one really good way. Not just the fact that this guy's dead, this guy's alive, but this guy is incredibly prosocial, so he gives tremendous amounts of his fortune to help other people. And this guy, as far as we can tell, was not very fun to be around and was not as giving. I've since learned that probably one of the major factors in this guy being so prosocial and charitable is his wife. It's the person he married, but still there's probably something fundamental about him that makes him open to giving away his money. So how do we account for that? Now, there's been a lot of interest in the past decade and a half or so in a number of different neuromodulatory systems that seem to be involved in regulating social behavior. Oxytocin is probably foremost among these. Oxytocin is a peptide hormone produced in the hypothalamus, which is involved in mammals in giving birth, in lactation. It's also involved in bonding between mother and infant, and we've learned from the work of Larry Young and Tom Insel that it's really important for pair bonding in different species of rodents. For example, voles that form strong pair bonds have high density of oxytocin receptors in their quote-unquote reward system, and you don't see that in vole species who do not form strong pair bonds, suggesting that it's basically the localization of receptors within reward systems that drives these differences in behavior. Kind of more critically, in the past few years, people have become very excited in the idea of using oxytocin as a potential therapy for a whole variety of disorders that are attended by social dysfunction. Principal among these, autism, a disorder characterized by low motivation to interact with others. This is just a result from a recent paper from Kevin Pelfrey's lab showing that when people with autism inhale oxytocin, which is thought to get oxytocin into the central nervous system, this can kind of reawaken parts of the brain that are involved in social perception and social behavior. So we have begun to try to understand how this all might work at a cellular and neural circuit level. And so we had decided to use the same kinds of delivery mechanisms that have been used in people, that is intranasal delivery, but now in monkeys. And monkeys, as you might be aware, they have to cooperate with us. They're not so interested in having liquids or sprays squirted up their noses. And so what we did was to capitalize on a pediatric nebulizer, which if any of you have young kids, you probably had the experience of your kid going to the emergency room if they had a bad cough and they would inhale some albuterol, which would open up their lungs. And so we train monkeys to accept a mask over their face like this. They're perfectly happy to do it. And then we aerosolize oxytocin for them to inhale. And we give them basically the same sort of calibrated dose that the humans are getting. And the nice thing is that we can do cerebrospinal fluid draws from the cervical spinal cord and show that when monkeys inhale, this oxytocin actually gets into their central nervous system. Okay, that's the first demonstration that that technique actually does so. When we look at our reward allocation task, that is when monkeys are choosing whether or not to give a reward to another monkey, I'll draw your attention here. This is that preference index or vicarious reinforcement index, and what we're comparing here in gray and red is inhaling saline or inhaling oxytocin, and this is about 30 minutes after giving the treatment, and we see that initially there's not much difference, but over time across the session we see this incredible linear increase in the preference to give reward to the other monkey. That is an increase in prosocial behavior, and this is also accompanied by an increase in our donor monkeys paying attention to the other monkeys. So they're looking at the other monkey, they're watching him drink the juice, and it doesn't take a rocket scientist to imagine that perhaps all you have to do to increase or enhance social interactions, positive social interactions, is just get the monkeys to open up to each other, put down their normal wariness, spend more time looking at each other, and that attention to the other individual can increase the internal reward process through a kind of feedback loop. More recently, we've begun to try to tie this all together by giving monkeys oxytocin while we have electrodes in various parts of the brain, so we can look at what happens to neuronal activity as a result of oxytocin coursing through their brains. And this is not even worth showing you, but it's just sort of a schematic of what we might hope to find, although this is real data. If we compare kind of pre-treatment to post-treatment, the neuronal activity in the anterior cingulate gyrus becomes higher fidelity, so there's less noise and more signal. And we had already demonstrated previously that under those conditions, monkeys are more prosocial. And we don't see that with saline. So this is very, very preliminary. And we're also looking at kind of tying this together in one more way, which is to locally inject oxytocin in these areas and see what it does to neuronal activity and see what it does to behavior. Now, finally, in the last couple of minutes, I want to get to that last point, which is one about, okay, so we've looked at neuromodulatory systems, and that can have an influence on social decisions, but how does what an individual is born with, their biological endowment, actually shape their behavior? Now, we heard a lot today about the heredity of various kinds of traits, and we think that it's probably quite high for certain kinds of behaviors. We think that's probably true for a lot of social behaviors. Trying to dissect the contributions of genes and environment to the behaviors that I've described today is very difficult in a laboratory where you have, say, 12 monkeys. Okay, it's virtually impossible. And so what we have done is to abandon that approach. We've abandoned a lot of things in this study, but we've embraced spontaneously occurring variation in genes and behavior. So what we're basically doing is we are studying the patterns of behavior, both naturally occurring and in response to experimental challenges, amongst a population of about 1,200 rhesus macaques that are on an island off the coast of Puerto Rico in the Caribbean. They were deposited there 75 years ago. There were 400 founding members of this colony, brought there from India, and it's basically been a free breeding experiment. So they're very well habituated. We have exquisitely detailed records on these animals, their pedigrees, their life history, etc. They're completely habituated. And then about once a year for 6 to 8 weeks, we trap the monkeys and we collect all kinds of biological specimens, including blood and other measurements. We have a small laboratory down there. We spin out DNA. We ship all this stuff back to Duke. And we've begun to look at the relationships between the genes that these monkeys carry with them and ultimately their behavioral decisions. And just sort of as a promissory note, kind of to end on here, this is from a paper that we published last year in which we used social network analysis, this is a graph theory analysis, to look at the relationships amongst all of the monkeys within one particular group. So there are about anywhere between 9 and 12 groups of monkeys on this island at any one time. They kind of come together and they split apart. This is based on observations over the course of about two years. So each monkey was followed for, I can't even remember now, a couple of hours over the course of that time. And the circles are males and the squares are females. And this is based on positive interactions, so basically monkeys grooming each other. Okay, it's like hanging out with your friends. And so those monkeys who are in the center are at the center of everything. So they're the most popular. They have the most connections, which are indicated by these lines with other individuals. They have the most friends and the most allies. Monkeys here out on the periphery, they might only have one connection. That means they have one friend, and that's it. These monkeys here have no friends. They're absolutely friendless. It's really sad. Now, what I've mapped onto here in color are just the alleles for two genes that are involved in regulating serotonin function that have been of interest for humans. One is the serotonin transporter promoter polymorphism, and the other is TPH2, which regulates the production of serotonin. And so the monkeys in red have kind of the double whammy of the minor alleles. That is, sort of think of it as the low-functioning alleles for these two genes. And you can see that statistically they're more likely to be on the outskirts of the group to have fewer connections. In fact, they have statistically fewer connections than our monkeys who are homozygous for the major allele of both of these genes. Now, this is only one. I don't want you to come away with this thinking that I've explained all the social behavior in these animals. It's clearly much more complex than this, but it begins to show us how we can use a system like this in which it's the exact opposite of how you would approach the question in a laboratory. So these monkeys, they have a completely heterogeneous genetic background. They come from completely heterogeneous experiences and environments, but through painstaking observation and the very detailed records that we have, we can begin to try to piece these things together. And ultimately, we imagine that we can also select monkeys based on their genotype and their phenotype and bring them back to the laboratory to try to understand these processes. So just sort of to sum up, the primate evolutionary toolkit includes neural circuits for both empathy and strategic behavior, and neuromodulatory systems like oxytocin, serotonin seem to regulate these decisions, and this may happen partially by genes and the way they set up these systems during development. I want to convince you that primates are a powerful model for understanding the basic biology of decisions, and in particular, we might be able to capitalize on naturally occurring biological and behavioral variation as a powerful translational tool. And these are the folks who did all the work and our funding. So thanks for your attention. I'll take some questions if you have time. I don't know if we have time. Yeah, Mike.
Just as a general question.
Yep.
Just from your experience, trying to look more natural.
Yeah. Yeah.
So these things that they say that monkeys or primates can't do, we talked about the
Yeah.
neuroeconomics test
Yeah.
or empathy, or it looked like some
Yeah.
very good observational learning there.
Yeah.
How much is it they just don't get it because he's up in the laboratory that is so abstract, away from the natural environment?
Well, I think,
versus-
yeah. Well, I think that we have to think much more like monks. So the question was, so a lot of, there's often a bias in many fields to think that animals can't do or can't express a variety of the kinds of behaviors that we do. And I think that we have to be much more open to the possibility that they do, and to the extent that, if we can incorporate the kinds of naturally occurring information processing problems in the laboratory, then they're much more likely to express those behaviors. I mean, it's interesting to us the difference in training a monkey to perform the kinds of tasks that I showed you here, or foraging tasks like you described earlier, compared to, say, making a very fine threshold perceptual discrimination. That's very difficult to train a monkey on because you're connecting an arbitrary stimulus with an arbitrary response and outcome. So is the line oriented, slightly this way? Move your eyes to the right and juice will come into your mouth. That's very strange. If you ask monkeys to perform these other kinds of actions, solve these other kinds of problems, they seem to tap into the kinds of problems they are equipped to solve. They're born to solve, if you will. So when we've given them foraging-like problems, they will approach the optimal solution within 1 or 2 days rather than 6 months. They can learn to often, Wei Song, my postdoc out there, is going to say that's not true, but I will say that in many cases when they are performing some of these social tasks, even learning to manipulate a joystick can happen relatively quickly. So my own personal bias is that we're underestimating the animals, and that our own, a lot of the capacities that we bring to the table and express, they probably do as well. They just may not be quite as developed and sophisticated.
One type of depressed monkey is caused by social defeat, and the other is caused by social isolation. So I want to know if there are any differences or similarities correlated with the brain areas you have just mentioned.
Yeah, I mean, I think that's a really good question. We have not looked at anything related to either social isolation or social defeat. The way that those are produced in the laboratory is very different from the way that an animal might experience some degree of social isolation or social defeat out in the real world. So, I mean, these monkeys are experiencing defeat all the time, right? But they usually still have someone else they can kick around. Isolation, even those monkeys who are don't have any friends, they don't have anyone to groom, they still get in fights with a lot of other monkeys, and they still can see a lot of other monkeys, so it's not truly isolation. So, I mean, these are really interesting questions. I have been generally not highly motivated to implement those kinds of studies in my laboratory just because that will be hard for me to do ethically. The question was, I think, is there any interaction between the dorsolateral prefrontal cortex and the anterior cingulate gyrus? I don't know of direct connections between those areas, and we certainly haven't looked for any kind of correlated activity or correlated response. But you're thinking in terms of if the anterior cingulate gyrus is important for thinking about the state of mind of the other monkey, and the dorsolateral prefrontal cortex is important for then planning behaviors in that social context, how does that transformation occur? That's a great question. That's difficult for us to answer given the kinds of methods we've used up till now, which have been, for the most part, serial recordings of single or multiple neurons. I think the next thing that we are tooling up to do is to put in kind of massive arrays of electrodes that will allow us to investigate what's happening in many areas at once and look at the timing and the correlations and the coherence between those areas, and then we'll be in a much better place to answer that question. Well, you know, that would be the end of PhD students in the United States. So, I mean, I joke with my graduate students that I could replace them with monkeys. They could do all the experiments, but that's not really true. So, I don't know there's no real evidence that monkeys engage in those kinds of behaviors where they sort of shepherd the behavior. You know, if you think of ants doing like aphid farming, you know, where they tend other species to get some reward back from that. And we don't see anything like that on the part of a monkey. But I don't know if movement can confer new capacities on these monkeys for self-awareness, then maybe they will, maybe they can start to do that.
Have you looked at how hunger or satiation affects the donor task?
Yeah, the question was about satiety. Does that have an impact? You might expect.
You're feeling generous?
Yeah, you're feeling generous because you've had a lot to drink. So we've not looked at it systematically in the sense of like varying the amount of juice or the amount of liquid that the donor monkey has had. We tend not to see big changes over the course of the session. You see a slight, maybe a slight decline in behavior, but it's not significant. Yeah, I don't think there's really anything there. The problem for us is that we worry about if you come in and you have them performing a task and you give the donor half a liter of water, he just might stop. I mean, he might stop performing. He might not. I'm always trying to get people in the lab to try that, but of course their time is more valuable than mine, so they don't want to take me up on that experiment.
The bullying question is, if the two monkeys are, one is the mother monkey, the other is her baby, could the mother sacrifice herself to let the baby drink more?
Yeah, I think this. So this is a question if there are other kinds of social relationships between the animals. We don't have any we don't have any mothers. We don't have breeding. We don't breed monkeys in our colony. But it's an interesting question as to what kinds of social relationships might shape this behavior. So we do have, we now have a lot of female monkeys in the colony, which we didn't have when we first started this, and so we have begun to look at whether male donors are more prosocial to female recipients, and anecdotally, that seems to be true, but that's not, I mean, that's not, you know, I would never state that in public, although I guess this is public. And we also can imagine, we can also look at whether females just generally are different in their patterns of prosociality. It's important to remember that female rhesus macaques are as competitive as males and as sort of status obsessed as males are.